Hello everyone, welcome to today's webinar. We are glad to have you here. Our speaker today is Dr. Tan, Senior Technical Expert from Vazyme. Today we'll talk about the presentation on RNA extraction and followed by a live Q&A session. Feel free to drop your questions in the chat box and we will address them at the end of the talk. Let's give a big welcome to Dr. Tan.
Thank you, Jane, for introducing me. Hi, everybody. So my name is Dr. Tan, or you can call me Jason as well. And I'm the technical support for the Vazyme group, okay, with technical expertise. So today, I'll be going to share you on how you can quickly obtain high-quality RNA. As we all know, genomic extraction is indispensable in molecular experiments and is the first step of all molecular experiments. However, during the extraction process, especially when it comes to RNA extraction, we often fail to achieve the desired concentration and purity. So today, the lecture aims to help you solve this problem. Okay, so there are four aspects that we'll be covering today. Firstly, we will understand why RNA degrades so easily. Secondly, how to obtain high quality RNA. Third is how to inspect the quality of the extracted RNA. And finally, addressing common issues pertaining to RNA. So first, let's delve deeper about RNA itself. What is it? What can you do about it? And why does it degrade so easily? So we need to understand first the composition and types of RNA before delving into RNA degradation. As the carrier of genes, RNA is responsible for transmitting genetic information from DNA to proteins. It is composed of three parts that you can see on the screen: ribose, nitrogenous bases such as A, U, C, G, and phosphate groups. These components are connected in a long chain by 3' phosphodiester bonds formed between the phosphate groups and hydroxyl groups of nucleotide. Most RNAs found in nature are usually single-stranded. Okay? RNA is incredibly diverse and can be broadly classified into two categories: mRNA and ncRNA. So the mRNA, or messenger RNA, contains vast amounts of genetic information, although it accounts for only a small percentage. It is currently the most studied type of RNA. Then you have the ncRNA, or we call it non-coding RNA, which encompasses a wide range of RNA types, such as RNA, rRNA, tRNA, snRNA, and RNAi. In addition to their roles at the transcription level, these ncRNAs also play a crucial regulatory roles in epigenetic gene regulation. Now, RNA has a widespread application in basic research, biotechnology, biomedicine, environmental science, and agriculture. In basic research, common studies would include genetic information transfer, gene expression, RNA sequencing, and RNA data analysis. In biotechnology, typical applications involve gene expression analysis, RNA synthesis, and modifications. In biomedical research, there are techniques such as RNAi, RNA vaccine, and RNA gene editing. The two most frequent used methods in scientific research and academia to investigate RNA are sequencing analysis and gene expression analysis. Now, sequencing analysis involves RNA extraction followed by library construction and sequencing analysis. Gene expression analysis examines gene expression levels through PCR after RNA extraction. Now, the quality of both analyses relies on the RNA sample itself. That means if the RNA used is degraded, it will affect downstream data analysis. So what causes RNA degradation, as you might ask? We can analyze this from both internal and an external factors point of view. First, with regard to internal factors, RNA itself is inherently fragile in nature as it's made up of long chain connected by 3' 5' phosphodiester bonds. Okay. Now, the free hydroxyl group, which is OH on RNA, that's easily attacked by nucleophilic reagents, leads to 2' 3' cyclic phosphate intermediate formation, therefore resulting in subsequent decomposition of RNA. It is therefore vital to use fresh samples or adopt appropriate preservation methods of RNA samples when conducting experiments. Okay. Now, on the other hand, external factors can also strongly interfere with RNA stability, and it is known that RNA's enzymes, which are widely available in various environments, has potent degrading effects on RNA. Now, it is difficult to remove it as it has extremely stable biological activity due to the disulfide bonds in its molecular structure, enabling it to withstand high temperatures and pressures. In summary, both of these factors contribute to the instability, vulnerability, and therefore degradation of RNA. We also learned that to avoid RNA degradation, fresh samples and mild reagents must be used in an RNase-free environment. So how do we obtain high-quality RNA? That might be the next question. First, we need to know what constitutes high-quality RNA. Intact RNA structure, or no residual impurities, and optimal RNA concentration. Now, to achieve these characteristics, extra attention is needed during extraction process. Let's look at some of the key considerations for each step. Firstly, let's look at sample collection and processing. In research, there's a vast variety of sample types to extract from, such as plants, animals, microorganisms, viruses, and more. As you can see on the slide, this is basically a different set of samples you can extract from, all of them which contains small amount or huge amounts of RNA, okay? And as you would suspect, total RNA content will vary slightly or significantly among different research subjects, or even with different parts of the same research subjects. You can see samples with RNA content, such as from heart and liver, can yield about 2 to 4 micrograms of RNA with 1 microgram of sample. Samples with low RNA content, on the other hand, like bone or corn leaves, okay, may only yield about 0.05 to 0.1 micrograms of RNA with 1 microgram with 1 milligram of sample input. So therefore, it is essential to understand the RNA content of your samples, how much it contains in there, and where is it coming from, before extracting or before performing extraction to assess the suitability of the resulting RNA concentration. As we know earlier that RNAs can significantly impact RNA, it is crucial to remember the following when collecting the RNA from samples. Immediately inactivate endogenous RNAs after tissue or cell death. I could not stress this any further, okay? Now, whether dealing with cells, tissues, or plants, or either directly lyse, immediately directly lyse or immediately extract the RNA, store the samples at -80 or with RNA preservatives. Okay, this is to help to extend the life of your RNA that you have extracted. Now, improper sample storage can lead to RNA degradation. This figure here on the slide shows RNA degradation in samples after 1 to 2 rounds of pre-store cycles and 5 to 10 minutes at room temperature. This is following the removal of the samples from -80 degrees. Okay, significant degradation can be seen in these samples, as you can see on the slide. All right. In summary, the key points of the sample storage are as follows. Ensure no RNase contamination by using RNase-free consumables throughout the experiment, and make sure you use fresh samples and appropriately store raw samples to prevent RNase activity. Now, moving on, second step is to perform sample lysis to release the genomic material from the sample. Okay. This step requires determining the appropriate sample volume, operating at low temperatures throughout, and ensuring complete lysis of your sample. There are 3 common lysis methods. First method is through enzymatic digestion by using Vazyme, cytolytic enzymes or proteases. Now, it also requires mechanical equipment. However, it's quite time-consuming. Second method is through mechanical lysis by using liquid nitrogen and homogenizers. They are efficient and suitable for difficult to lyse samples that can be quite intensive to the sample. Lastly, chemical methods which involve chaotropic salts and detergents. They are convenient, fast, and suitable for samples that can be easily lysed. So between these 3, you should choose the appropriate lysis method based on your sample type. So for example, plant materials with cell walls and abundant polysaccharides often require liquid nitrogen grinding. while fungi with thicker cell walls very need lysozyme for cell wall disruption and penetration. Animal tissues are commonly live using homogenization or liquid nitrogen grinding. Now, other than lysis, it is crucial to also inhibit RNase activity. The most common method involves utilizing strong oxidizing reagents to disrupt the tertiary structure of RNase, thereby inhibiting its activities such as guanidine salts. Triazole is often used as a mixture of guanidine isothiocyanate and phenol. Another option is to use RNase inhibitors such as reducing agents, which inhibit RNase activity by reducing the disulfide bonds in RNases. The last step, which is also most crucial, is the RNA extraction and purification. There have been significant advancements towards RNA extraction approaches since RNA was discovered. As you can see, around 1956, phenylchloroform extraction was initially the default method, despite its toxicity and time-consuming nature. Its high yield and effectiveness is still a popular choice among many researchers as of 2025. All right. Subsequently, column-based extraction was developed around the early 90s, offering simple operation and reduced physical impact compared to the organic extraction methods. Around this time as well, magnetic bead extraction method was then introduced, allowing high yield of RNA to be obtained with its simple operations. Now, these reagents can be easily superimposed into the extraction instruments, you can see on the slide as well, allowing extraction automation, leading to simpler operation and greater time savings. Now, all of these three methods mentioned above that are currently used in our research. So let's investigate deeper into each one of them. Okay, so firstly, to understand the organic and precipitation-based extraction, we must need to understand the purpose and function of each of the reagents. From left to right, we have phenol, which is a crucial component in triazole-based extraction reagents, effectively promotes protein denaturation, cell lysis, and the release of nucleic acids and proteins. Then we have guanidine salts, which is a potent protein denaturant, added to the reagents to dissolve proteins, disrupt cellular structures, and exhibit strong denaturing effects on RNases. Then we have chloroform, which is a heavy organic solvent that rapidly separates the organic and aqueous phases. It creates a three-layer solution with the upper aqueous phase containing primarily RNA, a middle white film layer, with DNA and proteins, and a lower organic phase consisting mainly of chloroform and denatured proteins. This phase separation facilitates subsequent RNA extraction and purification. Lastly, we have the alcohols, which are either ethanol or isopropanol, which compete with the nucleic acids for water molecules, therefore causing nucleic acids to dehydrate, aggregate, and precipitate, ultimately yielding your RNA precipitates that you're looking for. Okay, now the method of extraction is based on a principle using guanidine salt or phenol to lyse cells, followed by chloroform extraction to remove the impurities, and precipitation with isopropanol and or ethanol to obtain RNA. All right. Now, given the complexity of the triazole experimental process, there are a few considerations to take note of. Firstly, after using chloroform to separate the liquid into three layers, carefully separate and aspirate the upper aqueous phase containing the RNA as much as possible without aspirating the middle layer. After adding the alcohol, place the mixture in -20 would help to increase the yield. Now, for more complex samples and high impurity levels, repeating the extraction step or additional 75% ethanol rinsing steps might reduce the yield slightly, but it will help to significantly improve the RNA quality. Now, given the reagents such as phenol and chloroform are known to be hazardous chemicals in the laboratory and are toxic to end users in general, now our company has researched the latest technology and have come up with a safer substances which are alternatives to chloroform. As you can see in our slide, okay, the one above, we have used our own lysis buffer, okay, and this latest reagents not only maintains the extraction concentration and purity, but also reduce harm to end users as well. Okay, let's move on to the next method, which is the column extraction method, and this relies on specific absorption of RNA using centrifuge column containing common absorption materials such as silica-based materials and anion exchange resins. Silica-based materials possess the ability to significantly absorb nucleic acids, enabling them to efficiently capture RNA from complex biological samples. Nucleic acids bind under high salt and low pH concentrations, which basically you would have the sodium ion that acts as a bridge, absorbing negatively charged oxygen on the phosphate backbone of nucleic acids. In acidic conditions with high salts or high sodium, disrupts the hydrogen bonds between hydrogen in water and negatively charged oxygen ions on silica, allowing tight binding between nucleic acids and silica. Subsequently, nucleic acids are PE buffer or distilled water. Now, on the other hand, anion exchange resins operate under the opposite conditions, binding nucleic acids in low salt and high pH environments and eluding them in high salt and low pH conditions. The last method, which is shown in the slide here, is magnetic bead-based method. The interior of the magnetic beads is normally composed with polystyrene material, and the exterior is coated with hydroxyl molecules, which non-specifically bind to nucleic acids in the presence of salt and alcohol. The binding principle is consistent with that of the absorption column. Well, these reagents is normally used in magnetic bead-based method and can be integrated into automatic extraction instruments, okay, enabling full automated extraction and reducing labor costs. You can see in our instruments here on the right, and the operation is pretty straightforward as well. Now, simply add the sample into the plate-based reagent kit, place it in the automated instrument, and the extraction process will proceed automatically. Our company offers two types of instruments, either with the 32 or the 96 variants for selection. So, to summarize, if you have ample time and looking to save costs, the precipitation method would be the suitable choice. If you prioritize simplified operations, time saving and efficiency, the centrifugal column method would be the more appropriate option for you. However, you're working with large sample numbers, magnetic bead-based method combined with the automated extraction instrument would be the best option. So you have three options to choose between. Which one would be most suitable for your extraction? Okay. Now, once you have extracted your RNA, the next step is to assess the quality of the RNA. The following four methods that you see are commonly used for detection. So let's take a closer look at each one of them now. Now, one of the most frequent employed tools in the scientific research is the UV spectrophotometer, which quickly determines the concentration and purity of RNA using nanodrop or one drop. The detection principle relies on the fact that nucleic acids exhibit maximum absorbance of 260 nanometers, whereas proteins show their maximum absorbance at 280 nanometers. By measuring the absorbance peaks at different wavelengths, the RNA concentration can be calculated. This method is straightforward and convenient, but it may yield inaccurate values at low concentrations. As mentioned earlier, nucleic acids exhibit their maximum absorbance peak at 260 nanometers, while proteins do so at 280 nanometers. Therefore, when we say OD260, it normally represents the absorbance of nucleic acids. OD280 would represent the absorbance of proteins, and additionally, OD230 is used to assess the presence of contaminants in the sample, such as carbohydrates, phenol, salts, ions, and etc. Now, if the OD260 over 280 ratio falls within the range of 1.9 to 2.1, it indicates good RNA purity. Right? A value of 1.9 suggests an elevated OD280, indicative of residual protein contamination. Okay. Conversely, a value greater than 2.1 indicates an unusually high OD260, possibly due to RNA degradation. An OD260 over 230 ratio greater than 2.0 signifies accurate results, while a value less than 2.0 implies the presence of salt ions or contaminants from reagents. Now, it's worth noting that even though the OD results are not optimal, but the concentration is still decent, the RNA may still be usable downstream. If subsequent experiments have low impurity requirements, such as reverse transcription, quantitative PCR, or RT-qPCR, the experiment can still proceed. Okay? Many modern reverse transcription reagents exhibit strong tolerance to RNA impurities, and even some of our kits helps to remove any contaminants that might be still in there. All right, just to share. If the RNA concentration is low, or you desire a relatively precise concentration value, you can choose the fluorescent dye method, commonly known as qubit. This detection method involves fluorescent dyes that specifically bind to target molecules, such as double-stranded DNA, single-stranded DNA, RNA, or proteins, and emits fluorescence upon excitation by a specific wavelength of light. The instrument then receives the fluorescent signal and converts it into a concentration value, enabling precise quantification of DNA and RNA. While the cost of operation may be relatively complex, the resulting concentration values are most accurate. Now, assessing the integrity of RNA, let me just see here, okay. Agarose gel electrophoresis is frequently used, okay. Let me just go back one slide, apologies for that. Okay, to assess the integrity of RNA, the agarose gel electrophoresis is frequently used, right? RNA from both prokaryotic and eukaryotic cells typically forms 3 distinct bands in the gel. Now, if these 3 bands are clearly visible without smearing, it indicates good RNA integrity. Smearing or diffusion indicates RNA degradation, with more severe diffusion indicating more extensive degradation. The presence of bright bands in the wells after electrophoresis may suggest residual protein contamination, as you can see on the slide. It is crucial to clean the electrophoresis tank thoroughly and replace the electrophoresis buffer with fresh solution before running the gel. Otherwise, the presence of RNases in the electrophoresis apparatus can also lead to RNA degradation during electrophoresis, affecting accuracy of the results. Okay, now we come to this slide. For a digital assessment of RNA integrity, purity, or degradation level, the 2100 bioanalyzer is normally used. Now, the RIN, or also known as the RNA integrity number value, from the results can be used to judge the RNA integrity. Okay, the RIN ranges normally from 1 to 10, with higher numbers indicating higher RNA integrity and better quality. Right, a RIN value greater than 5, okay, indicates decent RNA integrity, while a value greater than 8 suggests nearly perfect RNA. So we always try to look for RNA RIN values 7, 8, and above. All right. Now you can see on the slide here. So the 4 different methods that I mentioned are the commonly used methods for RNA detection, and between the 4, you can choose the most suitable method based on your needs. All right. So during RNA extraction, you may encounter issues such as low RNA concentration, poor purity, and low integrity, or even failure to obtain the desired high RNA quality. How can we address these issues then? Okay, so let's just understand first that with regards to RNA degradation during extraction, the sample may minus 80 degrees. Secondly, improper sample pretreatment or RNase contamination may also be the cause. Ensure adequate pre-treatment using appropriate methods, such as liquid nitrogen or homogenization. Paying attention to the duration and temperature of the process as well. All right. Now, avoid overloading the sample, as excessive amounts may hinder the effectiveness of lysis buffer and RNase inhibitors. Additionally, ensure all reagents and equipment are free of RNase contamination by using masks, clean gloves, okay, and then operating in a dedicated clean area. All right. Just note that improper RNA storage or agarose gel electrophoresis can also contribute to degradation. All right. For RNA storage, aliquot and store at minus 80 for long-term preservation, or minus 20 degrees for short-term use. Aiming for prompt usage to avoid repeated freeze-thaw cycles, right? Before electrophoresis, treat the electrophoresis tag by soaking it in 3% hydrogen peroxide for 20 minutes, followed by rinsing with RNase-free DD water. All right. Now, ensure that the electrophoresis buffer is also prepared with RNase-free DD water. That's important. All right. Optionally, denature the RNA by heating it at 70 degrees for 2 minutes before electrophoresis to unfold secondary structures and ensure uniform mitigation when you run the gel. All right. Now, the second common issue is normally obtaining low RNA yield during extraction. Okay. So now, firstly, the sample itself may have low RNA content. All right. So some of the solutions would include understanding the RNA content of your sample and selecting those with abundant RNA. As I mentioned in the previous few slides, some samples might have high content of RNA, some might have low content RNA. So different samples will have different amounts. All right. Second is that you want to extract multiple tubes and combine to the dissolved RNA if you require. Okay. Adding precipitation aids the glycogen. So adding precipitation is like glycogen. All right. It will help in RNA precipitation as well. All right. So these are the three. All right. So improper sample storage or pre-treatment can also be a factor. Now, ensure fresh samples or samples frozen in liquid nitrogen and store it at -80 degrees of liquid nitrogen are used. All right. Additionally, you should ensure thorough sample disruption through homogenization or grinding before you start. All right. Now, lastly, is that insufficient elution of RNA during column-based extraction could occur. All right. If that happens, you would preheat the elution buffer to 55 to 70 degrees. Ensure it is dispensed directly into the center of the column membrane. All right. So then you will want to also extend the room temperature incubation time and perform a second elution after centrifugation if required. All right. Now, the third common issue would be you having gDNA or genomic DNA contamination in RNA extracts. All right. So if you face that problem, that could be due to excessive starting material. All right. So what you could do is that you could reduce the initial sample input or use DNase 1 for on-column digestion or purify the product with DNase 1. Right. Inappropriate centrifugation temperature during triazole extraction could also be a factor. As this triazole is partially miscible with water at room temperature, chloroform extraction should be performed, okay, at 2 to 8 degrees with high-speed centrifugation, right? Another reason also could be that taking too much supernatant during triazole extraction, which then can lead to contamination, right? So if that's the situation, reduce the volume of supernatant taken during the phase separation to avoid aspirating the interface and organic phase. Okay, so this is where our company will come in, and this is some of the solutions that we can help. So our company supports RNA extraction in several ways, right? So let me just briefly talk about our company. So Vazyme is a biotechnology company that's engaged in technological research and product development based on biochemistry, molecular biology, immunology, enzymology, chemistry, molecular engineering, and bioinformatics. And our protein engineering-driven product development platform, all right, high performance antibody discovery platform, which is based on the single B cell, multi-system recombination protein preparation platform, and our quantum dot modified coupling and multi-index strand expression technology platform. Okay, so these are the five our core innovation platforms, enabling us to swiftly develop life science, in vitro diagnostics, and biomedicine products in response to market demands. Right. Building on these robust platforms, we have been able to provide comprehensive services to a wide range of customers. As a result, we have provided services to more than 18,000 PIs in universities and institutes, okay, across the world. And our customers have published over 60,000 research papers using our kits as our solutions. We have cooperated with more than 5,000 enterprises globally as well. Okay, so as a result, through continuous technological innovation and product updates, we can meet the needs of our customers in various fields by offering the current product lines. As you can see on the slide here, we have 9 different product lines, which are nucleic acid extraction, molecular biology, cell protein research, next generation sequencing, biomedicine tools, and components instruments, lab consumables, enable disease detection, and IVD reagents. Okay, so based on the various methods used for RNA extraction and what we have gone through today, we have and we offer a range of corresponding reagents, including those tailored for automated systems as well. Okay, you can see on the slides here. Among our column-based extraction reagents, 𝗓𝗏112, all right, which is our star product, boasts not only rapid extraction speed, but also low toxicity as well. If you're considering precipitation-based extraction methods, our R711, which is a more user-friendly alternative to traditional trisol, eliminates the need for chloroform, therefore minimizing potential harm to users. All right. Now, in our automated extraction solutions, we provide specific reagents, as you can see on the slides, okay, for different sample types as well, like for cell and tissue, animal tissue and plants, okay, even for viral and also microbial extraction as well, okay, and basically tailored for different sample cell types. Now, every one of you in this call, all right, can choose the most suitable reagent based on their individual needs. All right. And in the research and development for extraction reagents, we have invested significant human and material resources as well. So through the years of research and refinement, we have established 4 key extraction technology platforms, okay, creating technical barriers that ensure stable output of nucleic acid extraction kits, thereby supporting the success of scientific research experiments. Okay, so the platforms include the 2 over here, which is silica membrane nucleic acid extraction technology, magnetic bead-based nucleic acid extraction technology, precipitation-based nucleic acid extraction technology, and nucleic acid degradation prevention technology. Right, so these are the 4 that we have. There's a lot of details right here, but if you're interested to know more about our technology platforms, you can always approach us at the end or even after this session. All right. So with that in mind, that concludes our presentation today. Once again, thank you all for participating, and I'll take your questions.
Thank you, Dr. Tan, for the informative session. Anyone with questions right now could raise your hand so I can turn on your microphone. Let's wait for like 1 to 2 minutes to see if we got some questions. Or you're welcome to leave your questions in the Q&A box. We'll answer them accordingly. Okay, it looks like we don't have questions at the moment. That's perfectly fine. If you got any questions later on, feel free to reach out to us after the session. And that brings to the end of today's webinar. Thank you all for joining, and a big thank to you, Dr. Tan, for the excellent presentation. We look forward to seeing you in our future webinars. Have a great day, guys.
Thank you, everybody. Have a good one.